I will talk about these other functional domain of 30S subunit. That let's suppose of course you know that these were the, what was these areas for? Yes, my friends, initiation factors, binding areas, right? Now what are these three pockets? What is this pocket? What is this pocket? And what is this pocket? These three domains. Of course 16 S's present over here, and here was special point which will bind the messenger RNA. What are these pockets? Actually these pockets bind the transfer RNA. These three pockets are transfer RNA binders, right? Which we will see later. But when transfer RNA are binding here, they arrive at this point. So this pocket is called arrival pocket, or we simply call it new transfer RNA. Suppose this is a transfer RNA which is coming here with an amino acid. So because it is loaded with amino acid, so it is amino acyl transfer RNA. Now, first pocket here, this will bind the incoming or newly arriving transfer RNA, right? So this pocket or binding point or domain of 30S subunit is called A point, A site. On 30S there is A site, but here I want to specify specially that this A site is not only present, okay, this is later on I will show you when 50S bind here later on, not now. Actually A site is partially on 30S and partially on 50S. So all this is A site. A site of 30S, A site in the 50S. What is A site? This is for arrival of, A for arrival of, yes, arrival of, what is this? Amino acyl transfer RNA. Amino acyl transfer RNA. So actually we call this A site because of amino acyl transfer RNA binding site. Most of the time this is for the arrival of newly coming amino acyl transfer RNA, right? Then the second pocket, this also partially extend on 30S and partially extend on 50S subunit, right? Slightly on 30S and large component on 50S. Here the amino acid which initiate the peptide formation, or sorry, transfer RNA which initiate at this point. What is here? Here is a transfer RNA binding site. Which transfer RNA? Either the transfer RNA which is the first one in initiating the peptide chain formation, or when peptide chain is being formed, that transfer RNA which is having the newly formed peptide chain, we'll see it later. The transfer RNA which is having, for example, this is transfer RNA here, right? And it is having, what is this? Newly formed peptide chain, right? This transfer RNA which is having few amino acids already, because peptide chain is being synthesized, as we will see later, this transfer RNA is called peptidyl transfer RNA. Now what is the difference between, this is coming here, what is the difference between this transfer RNA? Yes, this has one amino acid only, this is called, but incoming transfer RNA, this is called amino acyl transfer RNA. and this is called A site. Adjacent to that, there is another binding site where those transfer RNA bind which are either carrying the initiator amino acid, which we will discuss later, or they are carrying the newly formed peptide chain. So because this transfer RNA which is carrying the newly formed peptide chain, so this transfer RNA is called peptidyl transfer RNA, and this site is called P site. This is called P site. If this was A site for amino acid transfer RNA arrival, so this is P site. Why we call it P site? Because here is peptidyl transfer RNA binding. For us, we can also remember this is the parking site. Here is the arrival site. Here is the parking site where amino acid is either initiator, transfer RNA is there, or such transfer RNA is there which has newly peptide chain. And then we come over here. Here are usually, here was amino acyl transfer RNA binding, amino acyl transfer RNA. Here was transfer RNA with newly formed chain, right? And here is the transfer RNA which has given its amino acid to the peptide chain, and now it is about to leave. This was coming here, this is going to exit. Because this is going to exit, right? We call it E site. What we call it? E site. So what we see there, in the 30S ribosome, what we see there, that here were 3 special transfer RNA binding domain, right? One domain, then other domain, then another domain, right? Now this was called A site because it welcomes the upcoming or newly coming amino acyl transfer RNA. Then there is P site. This was arrival site. This is parking site, where such transfer RNA is present. Either it is going to initiate the process of translation, or it is that which is carrying a peptide chain. So P site. And here transfer RNA I have shown with one amino acid, right? Here is transfer RNA with newly formed, yes, peptide chain, peptidyl transfer RNA. What is it? Peptidyl transfer RNA. And here is the transfer RNA which is having, which has already given its, what is this? Amino acid. It is no more amino acyl transfer RNA. It is no more peptidyl transfer RNA. It is deacylated transfer RNA or empty transfer RNA. So A site is for amino acyl transfer RNA which receives the new, and P is parking place where transfer RNA with newly formed peptide chain or initiator transfer RNA is there, and this is for the exit point. So A site for amino acyl, P for peptidyl transfer RNA, and E for from where that transfer RNA is present which is about to exit the ribosomal system, right? Now, how many domains of 30S subunit we have discussed? Let's make a brief review. I said 30S subunit, number 1, it is the smaller subunit involved in translation process of prokaryotic protein synthesis, right? Number 1. Number 2, it consists of 16S ribosome and about 20 proteins, right? 16S ribosome and about 20 proteins. Functional domain, it has 3 transfer RNA binding site, A site to bind amino acyl transfer RNA, P site having the transfer RNA with peptidyl transfer RNA, and E site for deacylated or empty transfer RNA. And with on the flanks of that, on the flank of the A, there is binding site where initiation factor 1 is present, and on the flank of E site, on the side of the E site, there is binding site for initiator factor number 3. And this initiator factor number 3 and 1, they have many functions which I will discuss later, but one function over here is that they are preventing the 50s unit to bind the 30s unit prematurely, right?